All right, so let's get started. Thank you all for coming to the seminar, the first seminar, special seminar in the IQB seminar series. And it is my pleasure to welcome Colin Jackson from the Australian National University. Colin has been a professor at ANU since 2012, uh, and he grew up in New Zealand, got his undergraduate degree there, and then went to Australia to work with David Ollis, a crystallographer, and got his PhD with David. And then he went on to postdocs at Grenoble and at the Weizmann Institute. And his lab has done some really exciting and beautiful work on protein evolution and epistasis and things like that applied to proteins and enzymes and binding proteins and so on. So excited to welcome you here and look forward to your talk, Colin. Thank you for coming.
Thanks, Saga. I sort of invited myself, so thank you very much for graciously hosting me while I'm here in the US. But Saga came and he was our 2022 Craig Lecturer, which is a lecture award we have. So I met him then and I've wanted to come back and visit Rutgers ever since. So this is a new talk I've put together. There's quite a lot in it. So I'll try to get through it all, see how it goes. So basically what my lab is mainly focusing on and looking at now is trying to understand protein fitness landscapes for the protein engineering design field. And so the goal in our lab is efficient protein engineering. So I did a PhD in protein crystallography and directed evolution, and directed evolution is great, and there's some amazing papers and we've learned a lot from it, but it's not efficient. And there's a whole lot of projects that don't get published where people are just sort of like cranking a handle and generating variants and not really going anywhere. So that made me sort of swear never to do that again and focus on how can we make it more efficient? How can we reduce the library size from 100,000 to 100 so that you can actually test things properly? So this basically, the issue is a lot of these things start with a simplified view and an assumption, which is that protein engineering is a simple optimization problem. It's a hill climbing exercise on a smooth landscape where every step improves function. But the reality is that it really often fails. And one of the classic things is directed evolution often stalls. So in natural evolution, things keep evolving, but for whatever reason we do in a lab, after round 4, it might just not improve any further. So that's one thing that can happen. And if you try to do things rationally, nothing works. So you've got all these issues that are sort of cropping up, which tell me that our models and our prior assumptions aren't right. So the hypothesis is that the simple models are wrong. There's increasing evidence that protein evolution occurs on a rugged fitness landscape, although that's sort of just a qualitative, vague thing. But the idea would be if we can start to quantitate and understand ruggedness, then there's an opportunity to use this in terms of improving our protein design. So success requires us to navigate that. So this is where my lab is now in Canberra in Australia. It's the capital, but no one's ever heard of it. So it's a nice smooth hill, right? If you start at this side of the hill or that side of the hill, you can reach the top of the hill, right? It's like it would be great if protein engineering was like this. And this is where I grew up in the South Island of New Zealand. You can see it's very rugged. There's a valley. If you start here, you can't realistically get to there because you have to go through a valley, right? And you could get flooded and die. So this is an analogy for what we're trying to do here. So a smooth fitness landscape basically implies that the effects of the mutations are mostly additive. Any beneficial mutation is good, regardless of the sequence background. So you're going, these are good, and you just get better and better and better. If you start from here, you can reach the peak. So it's an assumption in most early protein engineering strategies. What it looks like it's more likely is like this. And this type of landscape explains a lot of other observations people have made in the molecular evolution field. So for example, what you see multiple peaks. The peaks aren't all the same height, okay? So this is what we would call a local maximum. So if you start here, you might reach this peak, right? But you're not going to get to this peak unless you do some extremely long neutral walk to this part of the landscape. Whereas if you start here, you could get to this peak, right? And there's peaks and valleys and peaks and valleys. So the next mutation may be deleterious depending on the sequence context. So this is what we call epistasis. And the success, depending on where you start in the sequence, is what we would call evolutionary contingency. So it's a historical contingency in the sense of like your future success is contingent on your history, right? It'd be like being a student to a degree, like your success in your PhD program is in some way contingent on your prior study. Okay. So the first, what I'll do is I'll go through a few examples. There's 3 or 4 examples that I'll talk through, which all sort of relate to each other. And then I'll end with a extremely ambitious unified theory of everything that still has a few holes, but I think is starting to come into coalesce, at least in my head. So this is a study we did a couple of years ago, although it was only published last year, with Batson Rahman's group, University of Wisconsin-Madison. And so again, there's a lot of computational things that are done, and these are great, although there's a lot of assumptions in purely computational work. So we wanted to empirically map a real landscape. So often there's a technique called ancestral sequence reconstruction. You can make a phylogenetic tree and you can reconstruct ancestors. And this was good in the sense that you can map a trajectory, right? But it's one trajectory on a tree which has hundreds of sequences. So there's a data issue here in the sense that you're looking at a fraction of the total thing. So what we wanted to do was to map a real landscape, a full landscape. So we looked at the Lac-I-Gal-A family of transcriptional repressors. This is like one of the most heavily studied protein families there are. The transcription factors, so this is a DNA binding domain. This binds to the DNA. This is linked to the ligand binding domain. The presence of ligand results in allosteric communication to the DNA binding domain, which causes it to bind or unbind from the DNA operator sequence, which is a specific cognate sequence that's recognized by the DNA binding domain. Fidelity is really important in regulation of genes. So for an enzyme, we see a lot of promiscuity. So enzyme A might catalyze 4 or 5 reactions at 0.1% of what it does. Its main reaction, this doesn't matter too much. But if you're talking about turning genes on and off, you don't really want a whole lot of crosstalk, because this is a much more important system to regulate tightly. So the interaction between the DNA binding domain and the operator, these are sort of co-evolving. So this is something that we wanted to look at, but we thought that this might lead to a significantly rugged landscape. So we made a tree, a big tree, 580 sequences and 577 ancestors, and we empirically made every DNA binding domain on the tree. So we made over 1,000 proteins that all have a different DNA binding domain sequence, and we fused them all to the same ligand binding domain of LAC-I. So we can just use lactose or a lactose analog to turn this on or off. We then just used, Vatson's group used a high throughput screening method using a GFP reporter system to look at whether they could affect transcription of the LAC operator. And so what we see when you look at the graph and you look at the tree is that colored here, you see enrichment is in red. So these are active DNA binding domains. It isn't the type of thing that you often see from an enzyme. So with an enzyme activity with this, you tend to see these would all be bright red, and it would be increasingly this red up to some ancestor. You see emergence of the activity and spread throughout the thing. What we're seeing here is that we're seeing it appear relatively sporadically across the tree in at least 3 different lineages, although it's only appearing in about 2% of the sequences. So it's a classically rugged landscape. It's there, it's not there, it's there, it's not there. You've got a whole lot of peaks here that are all distributed, and it's a rugged rather than a smooth Mount Fuji type progression. So we observed 32 different instances where function was either gained or lost between nodes. Okay, so this is 1 or 2 mutations are enough to turn it on or off. And again, this is a really extreme example of ruggedness, but it's not a gradual evolution at all. It's a series of rapid binary switches. So a single mutation can turn the function off or can turn the function on. So this indicates very high level of what we would call sign epistasis. So a mutation could be positive or it could be negative, depending on the sequence context that it's introduced into. We also see that of the active proteins, they're all quite different, right? So again, this isn't saying that there's one needle in a haystack that you have to find. There's at least 32 different needles. It's just that it's hard to find them in the sense of a rugged landscape around them. So there are multiple solutions, but the function is, the reviewers made us take metastable out of the paper because it has an actual mathematical sort of definition, but it's sort of a nice way to describe it in the sense of like, there's not a lot of robustness to this activity. This activity exists and it can disappear. Whereas again, what you see with enzyme often is that the activity could be robust to quite a few mutations before it's ablated and killed. So lots of different solutions to the problem, although they're all only marginally stable in terms of their response to additional mutation. So that's really interesting. So this is sort of the point where you're like, oh, that's really cool. We can see that there's interesting stuff happening. We can see ruggedness, but for me, it's more interesting to try to understand why you're seeing the ruggedness. So what are the molecular determinants? Because the structure defines the function, right? There's something about the structure, there's something about the system, which is making it behave like this, which is not a usual way for what we'd typically expect proteins to behave based on work that we've done with enzymes. So it looks like the best model we can come up with to explain the data relates to operator asymmetry, okay? So why is the landscape so rugged? The LGF repressors are dimers that must bind to asymmetric DNA operator half sites. They're actually tetramers and the DNA loops around, but it's essentially two separate dimers. So the key thing here is that the DNA operators typically exist as half sites. So they're palindromic sequences mostly, but they're not exactly symmetrical. So this side is different to this side. And so what this means is that this imposes quite a complex selective pressure. So instead of the protein evolving to bind a DNA sequence, you've got one protein because it's a homodimer. So one protein has to evolve to essentially bind two different sequences. And obviously this is hard, right? If you want to specialize at being a scientist, it's hard to also specialize at being a professional baseball player. Like there's not many people can do this. Or like I'm trying to get into baseball. Shohei Ohtani is rare because he's really good at pitching and hitting. Not many people can do this. Like this is a rugged landscape if you're looking at people like him. So it's the same thing here. Like the single sequence encodes a structure. This structure has to find a way to bind to this sequence and this sequence. And it can do that because you can adopt different conformations because proteins are conformation plastic. And it does do that. And this, for example, is an overlay of monomer 1 with monomer 2. You can see that the DNA binding residues are in very different conformations. So it's something that's achievable, but it's logical that this is a very hard thing to do and would exist sporadically across the protein sequence fitness landscape. So basically, if you look at this and test that hypothesis, the hypothesis would be a symmetrical operator should be easier to bind to. And that's exactly what we see, is that if we make a symmetrical operator, we get much higher binding affinity, okay? So it's the asymmetry in the operator that's making this a hard problem. So one sequence must bind to two DNA sequences. This is hard. Effectively, what you're looking at now then, in terms of why is it so rugged, is an interference issue in the sense of thinking about it in terms of protein fitness. Yep.
So if I understand correctly, that chart on the top right, you're showing the affinity of one protein sequence to the operator.
Yes.
Did you actually test whether the landscape is less rugged?
Yeah, if in a world of unlimited resources and time, that's the thing that we would have done. But it's one of these 3-year project things, and then you're kind of like, do we have another 3 years to do it? So that's the logical thing to do, would be to go back and take that 1,000 protein library and redo that with a symmetrical search. Yeah, good question, and something we absolutely would have loved to have been able to do. And so essentially what you've got is you've got an interference issue. So thinking about it conceptually, you've got one landscape for one half of the operator. sequence. You've got another landscape for the other half of the operator sequence. And so interpolating these two landscapes leads to a more rugged landscape. So because you get situations of constructive interference, which would be like positive sign epistasis, or negative interference, which would be like negative sign epistasis. And we see this a lot in complex systems, like, for example, wave fronts in the oceans. You get these big rogue waves and you get doldrums, and this is a common sort of thing. So it's starting to make sense in terms of the protein sequence fitness landscape, what's causing this ruggedness. And it looks like what a lot of ruggedness is coming from is that thinking about fitness as a holistic thing, really what you are doing is you've got a fitness landscape associated with protein stability, protein solubility, protein binding affinity, if it's an enzyme, catalytic activity. And then when you're starting to put all these landscapes together, there's only a few sequences that can do all 4 or 5 things. And you can imagine this gets more and more complex because there's more and more attributes that proteins need to have. So in summary, I was going to go through some boring summaries. So instead, I've got a wild speculation slide. So why? And to me, I think what this says to me is that actually, in this case, like for an enzyme, and this is a more complicated thing, and we're still trying to wrap my head around it in the sense of, has evolution evolved to be a certain way? So in this case, the operator sequence and the transcription factors are sort of evolving together, right? So one part of the question is how has the transcription factor evolved relative to the operator sequence? But the other thing that's happened is that these operator sequences have also evolved to be asymmetrical, right? So the system, if you combine the two, has evolved to be super rugged. Why would it evolve to be super rugged? Because this is less evolvable. You can't easily jump from one activity to another if something is rugged, whereas an enzyme can. And that, I think, is because regulation needs minimal crosstalk. So unlike catalysis, or especially like secondary metabolism, where things can be doing whatever, it doesn't really matter that much. It's all just icing on the sort of metabolic cake. Regulation needs minimal crosstalk. So it needs to be highly rugged, because you can't be doing half of your old job while you're doing your new job and already starting another job, right? And so ruggedness looks like, to me, it's a key feature of the evolution in the fitness landscape of transcription factors. And it's this asymmetry in the oligomerization that allows these rapid jumps in specificity. And so this can be conceptualized as overlaid interfering fitness landscapes. So we then wanted to look at this is a completely different story, the ligand binding domain. This is a much simpler function. So we would expect a smoother sort of landscape, but we don't have the resources to make 1,000 ligand binding domains because it would cost us $1 million. So we could do this because they're small enough, we could get oligos, and it still costs a lot, but it was more affordable. So fitness isn't just about binding affinity in terms of the Gibbs free energy binding. It's a combination of enthalpic and entropic components. And you can see there's a temperature dependence of the entropic term, which we'll get to in a second. So enthalpy, and this probably sounds like an undergrad lecture, enthalpy is things like hydrogen bonds and salt bridges. It's very easy to sort of understand conceptually. Entropy is not that hard. Like once you've got it, it's okay. But it's things like you release all the waters now have greater degrees of freedom. So there's an entropic benefit to displacing waters on binding, okay? And the other obvious one is like if you've got a protein and the apoprotein is very mobile, as the ligand binds and it gets stable, that's not good in terms of its binding entropy. So these two components interact with each other, and you get trade-offs and feedback. And so what we wanted to try to ask was, again, sort of a broader question of do proteins use the same thermodynamic mechanisms to bind similar ligands throughout evolution? So lots of people have looked at evolution like, are the binding affinity changes and the specificity changes? And that's cool. What I wanted to look at was at the thermodynamic level, do mechanisms change in terms of how these things work? So we took the same tree. In this case, we did the traditional way of doing it, which was we plotted one single trajectory from the common ancestor, ancestor 1, out to ancestor 4. We guess this is approximately 3 billion years ago, so it's one of the oldest protein families. So this is a long evolutionary pathway that we went through. And And so what we found is that actually this very, very, very distant ancestor, ancestor 1, doesn't bind lactose or lactose analogs. It binds a different molecule completely called D-fucose, whereas the affinity for lactose and lactose analogs increases along this trajectory. And we looked at them all, not just with binding affinity, but with a technique called isothermal titration calorimetry, which some of you might be familiar with. This is a way that you can thermodynamically measure the enthalpic and the entropic components of the binding. And so if you look at here, what you see in the top corner, this is the TM, or the thermostability of the protein. So you can see that the most ancient one is very, very stable, and we see a gradual decline in their thermostability. And this is consistent with what we understand of the Earth at the time. So back in the Archaeal, Precambrian times, things were very hot. And although things are heating up now, it's not heating up to that same degree. These are some of the sugars. So D-fucose is what we think the ancestral ligand. So one of the first ligands that bound to. And this makes sense in terms of all origin life stuff. It's a very simple sugar that would have been around at the time. And increasingly, eventually evolved to bind to lactose. In some of our screens, you'll see that we talk about BMDG. BMDG is used because it's an analog of lactose, and lactose binding proteins also bind BMDG. So what we see here, if you look at it, this is the KD, so lower is higher affinity. And so ANC1 starts off with some promiscuous binding of BMDG, relatively weak, and then progressively over an evolutionary trajectory, it ends up with a very high affinity binding contemporary LAC-I protein. So this is a classic story. Sort of makes sense. We get a specificity shift. We get mutations that arise that change the specificity, increase the affinity for previously what was a promiscuous binding event becomes optimized and becomes the primary binding. And we see the binding affinity for D-fucose is lost. So you get a trade-off because it can't bind two different things, because it's, as we just showed, two things at once is very hard. So it starts off, it's got good affinity for D-fucose. We couldn't measure 2 and 3 because it had nothing. 4 and LAC-I have very, very weak affinity. So this is sort of like a vestigial trait. It's like, you know how we all have like teeny little tails hidden under all our flesh? It's like that, right? So we've got like a vestigial binding of D-fucose. But the interesting thing, and I won't go into too much detail, is the binding mechanism is very different. So ancestor 4 binds BMDG with the classically enthalpic mechanism. So you can count the hydrogen bonds and add it all up. It all sort of makes sense. It binds based on it forming bonds with everything in the active site. Ancestor 1 binds D-fucose with an almost entirely entropically driven mechanism. So as well as a change and trade-off in terms of specificity and changes in affinity, what we're seeing is that the fundamental mechanism underlying binding the ligands is quite different. So being I'm not a former crystallographer. Being a crystallographer, I want to understand stuff like what's actually happening in the structures that underlies this. So we observe a dramatic shift in the thermodynamic strategy over billions of years. The ancient ancestor 1, the binding is entropically driven. Like I said, there's a temperature dependence to the entropy term, which is, I think, really important. So it relies on the hydrophobic effect, and then the modern LAC-I is enthalpically driven. What we see is exactly what we would not hope to see, but what we would expect to see based on the ITC. So if you look at BMDG bound in the active site of E. coli LAC-I, there's a whole lot of ordered water molecules, a lot of hydrogen bonds. So it's entropically disfavored because there's a huge amount of order there, but the order is compensated for by the energetics of all the bonding that's taking place. Does that make sense? Right. Whereas in the D-fucose binding ancestor 1, we see very few ordered water molecules. So the water molecules that were in the binding site have been essentially freed and liberated, and they're floating out free, living their best lives with the other water molecules. So that's an entropic benefit. And if you look at the overall structure stability, when the ligand binds to E. coli LAC-I, the protein becomes more ordered, which is, again, the classic way of thinking about how this works. The ligand binds, you get more ordered around the ligand. But D-fucose, this is a crazy structure. I've never solved anything like it. It was a 1.2 angstrom structure, but this domain looked like 1.2 angstroms, this domain looked like 3 angstroms. Like it's incredibly disordered. So even upon ligand binding, the protein's able to retain a huge amount of disorder to prevent that big entropic penalty of losing order upon binding the ligand. So this all makes sense, and this suggests that the mechanism of binding is itself an evolvable trait. And this sort of leads us to, again, sort of the wild speculation slide. A student made this figure. I just noticed on the y-axis, the temperature goes down to 0. It's not quite that cold. So I need to, it's a conceptual figure, but the temperature has gone down. And this is the thermostability of the variance, also goes down with the temperature on Earth, so it'll make sense. But what's quite cool is that in a hot environment, the entropic contribution, P(W)S) to free energy is magnified, right? So that actually makes sense that in this world where it's 80 degrees Celsius, entropically driven mechanisms make a lot more sense. And as things start to cool, the benefit that the protein can get in terms of binding from entropic processes like liberation of water diminishes. And so that imposes selective pressure to compensate for that. And so we have this sort of feedback, which means that there's a selective pressure to evolve enthalpically driven binding as the temperature term changes. And so that's sort of what we see. So basically, enthalpically favorable interactions like hydrogen bonds become more stable and are favored by selection. So the conclusion we got to is that the thermodynamic properties of the protein ligand evolution is probably itself shaped by environmental adaptation. All right, under the next one. So I've shown you a couple of ASR studies, and we do ASR a lot, and I think it's sort of useful, but obviously, like, it's a hard technique to do in terms of like you need to work hard to make the tree work, and you need to sort of, there's a lot of trial and error, and it's a little bit like artisanal baking, like I'm making a sourdough, but it takes 2 days. So obviously, has anyone heard of machine learning? It's a kind of a thing. So obviously we want to jump on the bandwagon because I need to get papers published and people get jobs. So we decided we're a machine learning group now. So we, with Nobu Tokariki from University of British Columbia, had done, well, his group did the directed evolution because I can't deal with it anymore. Our group solved a lot of structures, so we solved 20 or something structures along these different trajectories. So this is a bunch of papers. So we started off here at wild type. This is an enzyme called PTE, which is a phosphotriesterase, and you can evolve it into an aryl esterase. So you go from a tetrahedral to a planar substrate. It's still a hydrolase. And so this is like a 10 to the 5 change in activity going from the PTE here to the aryl esterase here. And so you can see here, this is essentially a graph of all the variants that have been made and characterized. And we've solved structures of all of the ones in gray. And so what you see here is that, you know, there's one trajectory that goes from here to there, and we've tested some pathways out from that. And then we evolved it backwards. So if you start from the aryl esterase variant and you say, we're going to flip the selection pressure again, go back to the wild type PTE, you can see that it doesn't go back the same way. So this was described by Joe Thornton's group as an epistatic ratchet. So essentially you're moving forward, you can't go back the same way. It's ratchet-like. I think it's maybe, you could also use the term hysteresis, right? In terms of chemistry and material science, like the process doesn't follow back the same way. And that will make sense later when we're talking about complex systems theory and stuff. So you get this hysteresis where you don't follow back the same way and you evolve essentially to the same activity, but a different sequence. And then here we blocked this trajectory with mutations and said, can it evolve a different solution? And that's our S trajectory. And then there's a whole lot of consensus site mutations. So a lot of papers, a lot of work over probably a decade. We've solved a bunch of structures. As an example, one of the first questions we had was like, what underpins this change in activity between these two? So there's a 10 to the 5-fold difference in activity. When we solved the structures, what we see, there was only one mutation in the active site. So how does And when that first mutation comes in, you get a 2-fold change in activity. You don't get a 10,000-fold change in activity. So how do all these remote mutations change the activity? And this, I'll get a whole thing about smoothness and ruggedness, but basically these remote mutations, they didn't change the ground state structure. So we solved all the structures. The final RMSD was less than 2 angstrom. So if you look at the ground state structures and you just superimpose them, it's all the same structure. And at that point, you're starting to lose your mind and coming up with insane theories. When you, in this case, we did ensemble refinement because we managed to get quite high resolution data for them all. You can see here that although the ground state structure, the average structure doesn't change, we get big changes in the displacements of the atoms. So initially in the wild type, this loop is very mobile. So this loop's open, and we'd studied it for 10 years before that, so we understand how the dynamics work. This is basically an opening and closing to allow substrate to get in and adopt a pre-organized state for the transition state stabilization and open again. These two loops are quite stable. And then by the end, that loop had completely rigidified, and it had rigidified to hold the initial active site mutation in place. And then these two loops had become flexible to basically compensate for that and take up that role. So it essentially worked by freezing out unproductive conformations. So it's a really nice example, and I'll start to use words like this and it will make sense by the end, of self-organization and the emergence of function. But anyway, back to the machine learning. The challenge is the relationship between sequence and fitness is complex and nonlinear because of the epistasis. So a machine learning model, particularly protein language models, can learn these complex patterns better than we can, faster than we can. But the problem is that they're computationally expensive and they're trained on global protein space. So I'm sort of the mind that I don't think information from an intrinsically disordered protein in the human brain tells us much about how a globular bacterial hydrolase works. In fact, it may actually make the model worse, right? So I think rather than breadth of protein sequences, what we really want is depth. You can't get more depth because there's not more depth in some cases. So this phosphotransferase is evolved very quickly, so there's not very many close homologs. But what we can do, you could see before with ancestral sequence reconstruction, we can reconstruct sequences. And we're not using the machine learning to reconstruct them. So it's not like this sort of feedback disaster you can get into with machine learning. These are all reconstructed with very careful statistical algorithms, and they all tend to behave very well. It's been, decades of work has sort of underpinned all this. So that made us realize that this is actually a really nice way to augment the data. So instead of augmenting it with more machine learning stuff, which is obviously not good, you can augment the machine learning models with reconstructed ancestral sequences. And there's also advantages to that. So basically, the other thing is that you don't just make one tree. When you're doing phylogenetics, this tree is statistically supported at this level, but there's a whole lot of other trees that are probably statistically supported at a similar level. So you can make hundreds of trees, and that allows you to generate thousands of ancestral sequences. And then you can make a much deeper language model around your protein, which it can learn from, because the nature of the data, and we'll show this, the nature of the data is also really important. If you give a learning model just a whole lot of, so basically here you see blue, riboflavin, bleach black, phlegm, flamingo. No one in their right mind would think these words are related to each other or how they could be related to each other, right? These are very different words. What's a flamingo got to do with bleach? If you go back to the Proto-Indo-European language, though, you can actually follow etymologically the progression and the evolution of language. Okay, does that make sense? So you can learn a lot more. It provides context, it provides new insights. You get to see all the epistatic interactions forming and changing. And this is a much more useful thing to learn from than, for example, just spreading sideways and pulling in more unrelated words, right? Which is, I think, the comparison between like a 150 billion sequence model or a 10,000 sequence model. That's 10,000 sequences that are all evolutionary related. So why does this work? So I was complaining to Saga, I'm having to learn maths as a 44-year-old, which is horrific. I don't wish that on anybody. So any students in the thing, you might not want to, but you should learn maths now while your brain is plastic and can change, and it will be far less and won't be like resistant to it. So we used a concept called Dirichlet energy, which is it's a measure of how rugged a function is, right? So if there's a lot of steep gradients, the Dirichlet energy will be high. If it's relatively smooth, it will be low. So basically, let's can look at how smooth the data is as it's been organized through the function. So what we see here is that the lower the Dirichlet energy, the better the predictive performance seems to be. And this seems like a fairly clear trend. And you can see here that the model we've made, this ancestral model called LAYS, has very low Dirichlet energy compared to other approaches, like just a one-hot encoding, for example, even with the same sequence size. So what we see then, if you sort of map the components with different embeddings, you can see here that this is ESM2. This clusters it tries to cluster the sequences, but it really is just sorting them by their sequence similarity. So it's a fragmented map, and it's really hard to learn from this how the different sequences relate to each other related to activity. Whereas you can see here that the LAYS landscape is much smoother. There's a much more connectedness in there. So this is essentially a graphical representation of what the Dirichlet energy is already showing us. So you're getting big sort of gradients here, and it's quite rugged, whereas this is a much smoother landscape. You can see you're going from low through to high. So you can imagine learning from this is much easier than trying to learn from that, right? So what does that mean? So basically this shows that the evolutionary relationship is really important because it provides context dependence. So LAYS performs better than other models on sparse data. The best comparison is this model we made, LEXSE. So this is the exact same number of sequences as the ancestral model, but they're all the closest related sequences in the database. So for example, the 10,000 closest sequences to PTE versus the 10,000 ancestrally reconstructed sequences. And you can see that the difference in terms of the R-square is massive. So it's 0.76 to 0.56. So again, when you look at this sort of graphically, how the sequence is clustered, the LAYS model has a very clear, smooth landscape. Yep.
Can I ask, like, what's the size difference between all?
Identical size.
They're all okay. So
Yeah. So ESM in terms of size compared to these ones, I forget how many sequences ESM2 has in it. 1.5 billion or something, like a lot. This only has 10,000, so it's a much smaller model too, which is another interesting quirk. I don't want to flex too much on that, but it's obviously very cheap. We can train it in-house. We don't need 1,000 GPUs to do it. So, but these two are exactly the same size. And you can see here again, this is a really fragmented sort of high Dirichlet energy landscape. So what we see is that reducing ruggedness and developing smooth models where things are highly interconnected allows better navigation and prediction because of the context awareness that they develop. So having done that, we then thought, can we apply this in any context? So this is confusing. I should have picked a different enzyme. So this is a PET-ASE. So the last one was PTE. This is PET. They're completely different enzymes, completely different substrates. This is one of these plastic degrading enzymes. So again, wanted to look at context dependence. We used what we now call multiplex ASR. So basically just make a lot of trees, generate a lot of variance. And you can see here, if you make a whole lot of ancestral variance, you're seeing activity appear relatively stochastically throughout that grouping. And you can then narrow down in the sequence landscape. So for example, 55 here, we then looked more closely around that patch in the sequence space, and we find better ones. So 55 started at 0.4, and then we found a bunch that were up to activity sort of 2 or 3-fold higher. So this allowed us to generate a lot of variance. And then how do you understand that? We use the Dirichlet energy and the T-SNE mapping just to show, again, it's a much smoother organization of the data with the ancestral model compared to a one-hot encoding here. And this then allowed us to find even better variants like these two. The interesting thing about this one here and this one here is that they're the result of combining two originally deleterious mutations. So what we found is that the in silico evolution we can do underpinned by these evolutionary models tends to find solutions that you'd never come up with otherwise. So they're great at looking at the context dependence and saying, this looks bad and this looks bad, but based on all this other stuff around there and this other stuff around here, we think if we combine them, it will be better. And so it's pulling up some of the really interesting ones. And so the model in the pipeline basically allows us to find non-obvious high performance variants. The last story, I think I've got time. Oh, I'll go through it quickly, is a bit of a change from like intense sort of lab math stuff to a fly. So we've discussed lab evolution, but obviously like this is only worth as much as the lab. No, nothing really, to be honest. So if you're making a protein that looks good in a test tube, don't try to say this is going to be great and it's going to solve a whole lot of human illnesses or environmental things, because it's not until you put things in their physiological context that you get to see stuff more clearly. And again, that is because you've got different fitness landscapes. Like the fitness is dependent on different things in different contexts and environments. So this is the Australian sheep blowfly, Lucilia caprina. While it evolved, organophosphate insecticides were introduced in the 1960s. Within about 7 years, people had noticed insecticide-resistant flies, and they could tell that the flies were actually breaking down the insecticide. So some sort of catalytic resistance had evolved. So that's really interesting. So we wanted to try to understand what was this neo-functionalization mutation. So neo-functionalization basically is like a new function has appeared that didn't exist. So some enzyme has changed function, neo-functionalized. So lots of work from our collaborators at CSIRO had identified the enzyme and the mutation. So it's this G137D mutation. It's essentially a serine hydrolase, and on the opposite side of the catalytic serine, which becomes phosphorylated here, an aspartic acid gets introduced. And this can then act as a general base, activate a water molecule, which can dephosphorylate at the serine and free it up after being inhibited to perform multiple turnovers. So the interesting thing that we noticed when we first looked at it, though, is that this has to be in one specific orientation to work. And initially, it can sample the productive conformation, but not often. Okay, so it seems okay, but not really optimized for its sequence context. So we wanted to look at this in terms of evolutionary contingency, because what we found is that some insects develop resistance very quickly, and others still have never developed resistance. We see this also in bacteria. So you hear about antimicrobial resistance. It's not every single microbe developing resistance. It's one developing resistance and then passing it around on a plasmid to all the others often, right? So some things seem to evolve better and more easily than others. Why is that? So we looked at 9 different species and we solved all their structures. And what we found is that the effect of the mutation is highly context dependent. So if you introduce this mutation in one genetic background, even though they're all orthologs, they all do the same native activity. If you introduce it in one context, it'll do something very different to another context. So here, this is just different time scales. This is 15 minutes, this is 60 seconds. But in blue and black, we have Lucilia caprina. So you can see going from the wild type in black, there's a burst because everything gets phosphorylated, and then there's the stationary inhibited phase. And the G137D mutation allows it to, after initial burst, perform steady state turnover. Whereas if you introduce the mutation into the Culex, which is a mosquito, it actually makes it worse. So you don't get a burst phase at all. It's obviously blocking up the active site, and you get a little bit of turnover, but it's very weak. Okay, so what we see basically is that if you look at the structures, the reason for this is the local environment where the mutation is introduced into. So if that local environment is compatible, it's good. If that local environment has leucine instead of methionine, you've got problems and it won't work. Okay, so it's a good example of evolution of contingency and shows that the path to resistance is contingent on certain historical mutations. So the other question we had is that, especially because it didn't look quite right when we solved the structure, can it get better? And that's also a question you can put into your grant applications and say, there's a reason to do this work because we can tell the farmers that resistance will keep going up, or we can say it's flatlined and there's no worries. Because they can still kill the flies, you just need to use 10 times more insecticide. So we use directed evolution to look at this. So here in red is the wild type, and then in blue is the first generation. And then we managed after 9 rounds to make a variant with 11 mutations. It was 1,000-fold faster. So miraculously great directed evolution experiment. The mutations, again, followed the same pattern as we saw in the previous example with PTE. So there was one active site mutation, and the rest were all radiated outwards. They're all remote mutations. The mechanisms we see are the same too, in the sense that they just change the conformational sampling of that active site residue. So it maximizes the productive state, or a better way of saying it would be freezes out the non-productive states. And then there's always the experiment that you don't really want to do, but you kind of should do and know you should do. So then we worked with some fly people, and we said, let's make some transgenic flies and see if this helps. And so what parameters are more important? So we made the flies, and you can see here that actually this 1,000-fold improved variant is about 30% less functional in the fly. Okay, so the LC50 was actually lower, and this sort of highlights a key disconnect here between the in vitro catalytic performance and the performance in vivo. This leads to the OP sponge hypothesis. So basically in the wild, the toxic level of insecticides about 1 or 2 micromolar. What we saw is that although we increased the activity 1,000-fold, we increased the KM about 20-fold. And so at these concentrations, the KM of the evolved enzyme is actually higher than the KM of the target acetylcholinesterase, whereas the G137D retains a KM that's lower than the acetylcholinesterase. So basically, it's more important for it to intercept it's like a, I guess in NFL, it's like a, I'm not even going to try. It's trying to intercept the insecticide before it can interact with this target and then slowly turn it over. This is okay, but it doesn't do anything if it's super fast, but more of the insecticides binding to the target before it can do anything. So it's just a good sort of a cautionary tale. So this now gets to, in the last 5 minutes, I'll go through a unifying framework, basically what we're looking at in terms of protein evolution, and thinking about it not so much as evolution as something that's special and unique, but thinking about it as, it's a multi-component system, there's lots of parameters, there's other systems like this. Are they all behaving the same way or not? Are all these special things that we think are special actually special, or in hindsight, are they all exactly how you'd expect it to behave if it's a complex system? So we see ruggedness, epistasis, contingency, emergent behaviors. Can we find a unifying framework for that? So protein evolution can be viewed through the lens of complex systems theory. And we're not the first people to sort of raise this, and it's been talked about in terms of chaos, which it doesn't really fit to because there's a specific definition of chaos, but it does sort of appear to be like a complex system. So this framework is used in information science to understand systems with many interacting components that exhibit emergent, unpredictable, nonlinear behaviors. And so if you sort of look at how proteins are starting to behave across, as we collect more and more and more data, and we're seeing the same things over and over again, we see self-organization, emergent properties in complex systems, and that's exactly what we see with proteins, right? So you see folds as oases of sort of structure and order across a structure sequence space, which is mostly disorder, right? We see turbulence. So turbulence is a common thing in complex systems. It's the same thing in going from fold A to fold B. There's no smooth path of everything structured between these two things, right? You've got a structured protein, 2 ordered proteins, and you can get from A to B, but you will go through a period of significant structural disorder to get there, and functional. Fractal geometry, the trees, classic fractal patterns, right? We're seeing the same thing because effectively in evolution, you've got this constant churn. The mutations don't stop. The mutations just keep going. Even if you're settled into a basin or what we would call an attractor of function, you keep evolving even in that basin, even around that attractor, you just keep circling it and circling it and circling it, and it never stops. So this is essentially like a time component in a way. So we get regions of sequence space, different attractors, different functions. The selective pressure changes, you create a new attractor like the phosphodiesterase. This attracts sequences towards it. And this is sort of like what you see here in terms of the sequence space. And this is a classic sort of attractor type phase space thing. I won't go into phase space, but we also see sensitivity to initial conditions and nonlinearity, classic behavior from a complex system. And we see that obviously like the starting point is basically half the issue with protein engineering project and adaptability and feedback. So we see this really nicely in that thermodynamic evolution thing, right? This is a clear feedback loop where as the entropic term is weakened by the declining temperature, the enthalpic term compensates for it and increases. So it looks like they're all sort of behaving in a similar way. So I'd like to thank a lot of really talented students in my lab, like Sasha Pulsford and Barnabas School and Dana Matthews and Matthew Spence and other students who have contributed to it. And we just talked about a few projects, but I've had some nice collaborations with Bats and Raman and Nobu Tokariki and John Oakshott in Australia, and obviously funding. So thanks very much. I hope my accent was, everyone, I hope has just been like, I didn't understand the word he said. So
Oh, it's
not too
perfectly
bad.
clear. Yeah. Okay. Questions for Colin?
Stunned silence. Okay, good.
No, well, that was really. Oh, there are questions online.
Chen Y. Mike, I see you raised your hand. If you could just put your question in the Q&A section, and we'll do Cheng Hua's. There's a lot of big words in this one, so
Okay,
it's not
sorry.
going to read that.
Yeah. Okay. The enthalpic
I
versus
can read it if you want.
maybe.
Yeah.
Okay, I just for the room, I'll just
Okay.
read it out. Yeah. The enthalpic versus entropic mutation discussion is interesting. If the thermodynamic conclusion stands, i.e., mutation is influenced by hot to cool environment, we would see greater enthalpic impact on the lab-generated mutation studies for most proteins since we are in the cool environment. Has this been examined and confirmed? Is there a readily available tool to decide whether a mutation is enthalpic or entropically driven?
That's an awesome question and something that we were sort of looking at and thinking about. And there is, I think, I can't say quantitatively. Qualitatively, we've been looking through stuff and it does seem to, enthalpy seems to be something that is more often addressed and appears to be more important in a lot of the contemporary changes and things that we see. At the moment, though, it's really hard to quantify it. But you know, my guess is, you know, if the world was 50 degrees hotter, we might be seeing mechanisms involving water displacement and stuff like this more often. So the great question, I don't know. I can't say other than speculate. Yeah.
Michael, do you want to ask your question? Okay. Okay, regardless, this is from Michael Manhart. Regarding the claim about ruggedness, ruggedness as an adaptation to reduce crosstalk, is there a way to compare this across species, especially eukaryotes versus prokaryotes, where there is likely big differences in the risk of crosstalk due to differences in chromatin structure?
Yeah, another awesome question. And again, these studies always suffer from the fact that they're sort of N of one, like the data we see makes sense in that experiment. But the real understanding, I think, will come when people do similar experiments in different organisms with different systems, and you can start to test hypotheses like that. So, and that's 100% right, because in eukaryotes, you've got entire other levels of organization. This may have reduced the selective pressure for fidelity, if that makes sense. So that's an awesome hypothesis and exactly what I would expect to see. Having said that, I haven't done it.
So I'll ask a question. So I'm interested in thinking about the two different types of entropically driven aspects, right? One is, of course, the hydrophobic effect or water displacement upon binding, and the other is conformational entropy. And thinking about that in terms of other fitness landscapes, like, for example, protein stability, right? Because, and the question is whether one might be more likely to arise at a hotter temperature versus the other, given that protein stability also requires, some rigidification, especially in hyperthermophilic proteins. People think that there are these salt bridges that, sort of decorate the surface. So what are your thoughts about the intersection of these two landscapes in terms of entropically driven phenomena?
If you allow me to wildly speculate again, I've
Of
got
course,
an idea
I
about
encourage
this.
you.
So I think a lot of the hyperthermophiles that we see now are recolonization of hot environments. So the thermal vents we see in the year 2025 probably aren't the same thermal vent that existed 4 billion years ago. And I think what we're seeing is, and this is like the hysteresis thing I talked about, like evolution can go in one way, and then to go back, you often go via a different way. So what we see in contemporary thermophiles is a lot of salt bridges, a lot of disulfide bonds, and these, I think, to me, are consistent with a post hoc patching together of a structure to be stable. When we make and they're mostly enthalpically driven processes. When we reconstruct an ancestor and that ancestor has a TM of 100, and you look at the structure, it doesn't have any traits of a modern thermophile. It's just really well packed and entropically extremely favorable. So I guess that sort of answers it. I think there's different solutions to thermostability, and I think those solutions reflect a hysteresis and the fact that it's probably, they were thermophilic, they evolved to become mesophilic, then they recolonized another thermophilic environment. And a lot of the paradigm around what makes a protein thermophilic is based on our contemporary understanding of what a modern thermophile looks like. But not, these thermophiles are different to those thermophiles 4 billion years ago. So
yeah, that's a great point. Yeah.
Wild speculation, but that's my.
What about the water? What about the water displacement? I mean, are some, right, like your observation is that the ancestral interface was drier and the modern interface is wetter in some sense. Could that be sort of tested in some way, on a larger scale by looking at reconstructed interfaces versus modern interfaces with similarly sized sugars, for instance?
Yeah, we've got review comments back, so we haven't a whole lot of might have been you. But no, there's a whole lot of things that we need to, again, like this is a really hard thing about this whole field, is that like you do something and it takes 4 years and it's interesting and it just creates, do it on 10 systems kind of. Yeah.
Are there any more questions?
One follow-up.
Colin, maybe you can just read it out, if you don't mind.
Also, regarding your finding about entropic versus enthalpic driven binding, is there a way to check this in extant thermophiles versus mesophiles? Yeah, that's exactly what we've been asked to do in the review comments, and we 100% will. But like I said about the extant thermophiles, I'm not 100% sure they'll behave exactly like the ancestral thermophile that we made. And that would be an interesting result. Either way, I think it's worth doing. It's a good question. It's also really the other thing that we should do, which we can't do, is we should do the ITC at 100 degrees. But you can, I mean, it's an equation. You can put the temperature up.
Gibbs cell
function.
modes.
Yeah,
Right,
yeah, it's a concept.
Yeah, that's right. All right. And if there are no further questions, let's thank Colin again for beautiful talk and exciting results.
Thank you.